All right. Hello, everyone. Welcome to this month's research seminar series called Creating New Brain Cells, a Look into Neurogenesis. My name is Alex Barber-Cross, and I'm a research program assistant and exercise instructor here at the BC Brain Wellness Program, and I'm very happy to serve as your MC for today's event. So neurogenesis is the process where new cells or neurons are created in your brain. This process happens into adulthood and can affect memory, mood, learning, decision-making, and even more cognitive processes. We have a very special guest here today, Dr. Jason Snyder from the Department of Psychology here at the University of British Columbia, who will walk us through the basics of neurogenesis, including how his lab investigates the role of neurogenesis in learning and memory and what they have learned so far. He will also share how the animal-based research done in his lab and the neurogenesis field might translate to humans, and how this might affect our understanding of disease and mental health. So before we get started, I'd first like to acknowledge that the BC Brain Wellness Program at the Djaved Mowafghian Centre for Brain Health is situated on the traditional, ancestral, and unceded territories of the Musqueam peoples. I'm joining you here from Vancouver, the traditional, ancestral, and unceded territories of the Musqueam, Squamish, and Tsleil-Waututh nations. As all of you are joining from different places today, I encourage us all to take a moment to reflect and give thanks to the lands with which we are privileged to work, learn, and play from. We have a couple of housekeeping notes before we begin. So if you have any questions for Dr. Snyder today, please type them in the chat box. There will be an option to ask questions directly to the person titled Ask Questions here, and we will try to answer potentially some of them through the session or all of the questions at the end of the session. So to keep everything streamlined, only the presenters and the MC will be unmuted for today's session. And lastly, this session is being recorded, and the recording will be available to you at our website at bcbrainwellness.ca in about a month's time, if you'd like to rewatch or share this seminar with friends and family. I also want to quickly highlight that today is UBC Giving Day. So this is a very fun fundraising opportunity for our program, and we hope you will all join in. So all donations, big and small, add up to make a huge difference. And we have several matching donors, so your donation will be double the impact. UBC has multiple challenges that can unlock additional funds to our program, like the Ultimate Supporter Challenge, which is where the cause supported by the most donors on April 5th will receive $5,000. Or the BC Brain Wellness 5 p.m. challenge, where if we have 50 donors give by 5 p.m. on April 5th, today, a gift of $10,000 from an anonymous donor will be unlocked. Our website has a banner at the top of the homepage with a direct link to the donation page, and I'm also going to send a link in the chat shortly. We're off to a great start already and have already raised over $19,000 with 94 generous donors. So we have achieved both our 10 a.m. and 5 p.m. challenges, unlocking an additional $12,500 thanks to our donors and matching donors. And we are hoping to raise $50,000 by the end of the day. Without further ado, I would like to move on to introducing our wonderful guest, Dr. Jason Snyder. Jason joined UBC in 2013 after attaining a PhD in neurophysiology and behavioral neuroscience at the University of Toronto. And he now works as a professor in the Department of Psychology here at the University of British Columbia, and currently leads the Snyder Lab, a neuroscience lab in the Department of Psychology and located here at the Djaved Mowafghian Centre for Brain Health. His lab studies neurogenesis through the lifespan, aiming to use neurogenesis to aid in the understanding of the biology of memory and mood. And Jason has led and been involved in research investigating sex differences in neurogenesis, the impact of neurogenesis on choice, decision-making, and memory, effects of exercise and stress on neurogenesis, and many more active and very novel areas of research, some of which he will be sharing with us today. So welcome to the Brain Wellness Program, Jason, and I will pass it over to you.
Thank you so much, Alex, and to the whole Brain Wellness Program for having me. You know, I love doing neuroscience research, and we're always thinking about how can we, you know, ask questions and do research that is going to ultimately benefit society. And it's a real treat, and you know, I feel really fortunate to be able to speak to a bit of a different audience than I'm used to today. So I think it's really helpful for me and inspiring, and I hope you guys enjoy this. So let's get started, and I will share my screen and put on my presentation. Here we go. So I've tweaked my title a little bit, made it a little playful. I've changed it to everything you always wanted to know about neurogenesis and neuroplasticity, but were afraid to ask. It may not cover everything you always wanted to know, I'll be honest. There's far more out there than we can cover today. But I wanted to go into a bit of the basics of neuroplasticity and why it's important, why it's such an exciting phenomenon in recent years. And from that, we can talk about the production of new brain cells in the adult brain. And so let's move forward. So what is neuroplasticity? Neuroplasticity is simply the structural and physiological changes in the brain which support flexible behavior. It's the idea that the brain is ever-changing structure that is not static, but can change in response to experience and allow for alterations in your behavior in a way that can benefit you, benefit others, benefit society. And so it's worth taking a step back and just asking ourselves, like, why is neuroplasticity a thing? And I don't mean like, why does neuroplasticity exist? You'll have to ask the heavens for that sort of answer. It just does. But why is it exciting? Why has it been controversial? And the reason, I think there's probably a lot of reasons, but one I thought of is, for many years, people thought of brains essentially like computers. We can add, we can subtract. We created these machines. Here's a picture of a Commodore 128. Maybe some of you had this back in the 80s. A computer that can perform some similar functions as to what we can do with our brains. And so even this advertisement says, thanks for the memory. It talks about a computer that can that has intelligence that can match your own, and so it's likening the computer's abilities to that of our brain. And while there certainly are similarities, it sort of led to this view that a computer's memory is kind of like our own. That memories, for example, on memory data stored on a computer is a perfect and stable representation of data. And that led, and that sort of, I think, probably bolstered some of the ideas that, well, that's what memories are in our brain. I saw this happen. That's the way it's always been. I can recall it. It's a fact. And it turns out that actually the brain is much, memory serves a bit of a different purpose for us. And it may not be as veritable and a carbon copy of what actually happened. And the reason for that is due in part to plasticity, the fact that the brain is ever changing, and in fact, memories are ever changing as well. So the brain can do some things like a computer, but it is not identical in its functions. And there was also evidence to suggest that the brain is somewhat fixed. Do we have a question? I hear a bit of sound. There was also some idea that the brain is fixed and not plastic simply because of early pioneering work on neuroanatomy. And so here we have a quote from Santiago Ramon y Cajal, a Spanish neuroscientist who won the Nobel Prize. His work 100 years ago led him to state, once development was ended, the fonts of growth and regeneration of axons and dendrites dried up irrevocably. In the adult centers, the neural paths are something fixed and immutable. Everything may die, nothing may be regenerated. So this is a pretty harsh decree, but is certainly valid to some extent. Obviously, the infant brain is growing and developing. We see children, their ability to learn is no doubt due in large part to the tremendous plasticity that they have. And relative to that, there is less plasticity in adulthood, and that's really what he was observing, except he, of course, due to limited methods, wasn't able to observe, the true extent of plasticity in adulthood. Likewise, there was another pioneering developmental neuroscientist named Pascal Rakish, who is well known for his work on studying the development of the primate brain. Specifically, what we're going to talk a bit about today, the process of neurogenesis, the birth of new brain cells. And he writes, the large, synaptically interconnected adult brain may not normally permit redistribution of new neurons and subsequent growth of axons. Axons are what neurons send to connect with other brain cells to distant destinations. Indeed, post-developmental neurogenesis is most prominent in species having a considerable capacity for axon regeneration, such as fish and amphibians, or in the avian brain, with particular seasonal changes in brain structures related to song production. However, the brain of primates, as well as some other species, may be uniquely specialized in lacking the capacity for neuronal production once it reaches the adult stage. One can speculate that a prolonged period of interaction with the environment, as pronounced as it is in all primates, especially humans, requires a stable set of neurons to retain acquired experiences in the pattern of their synaptic connectivity. And so the synaptic connectivity is just the connectivity between neurons, the networks that they create. And so essentially he's making the point that humans live a long time, and we need to remember things over extended periods of time. And so if we think that memory and information needs to be remembered in a stable way to be used for many years and decades, well, the brain is going to have to be stable as well, right? We know that memories are represented in the brain, so those representations of experience are also going to have to be stable. And so this is why he is saying the brain cannot grow new neurons in adulthood. Maybe other animals can, like fish or amphibians, but hey, there's just lower animals. And so to some extent, this was reasonable. This was 1985. Again, people are thinking, well, for data to be stored on a computer, you need a hard drive that can store all those bits and ones and zeros in a stable fashion. Well, maybe the brain is the same way. And we can look at some of the beautiful drawings of Ramon Cajal, drawing individual neurons. We see a couple examples on the left and in the middle, just showing their extremely dense projections that they have that receive inputs from other neurons. And Cajal was not just famous for drawing individual neurons, but on the right, we have a picture of the brain structure that we're going to dive into today, which is called the hippocampus. And this is the part of the brain that is responsible for memories for everyday events and experiences. What you usually think of when you think of a memory, like what you did yesterday, for example. And in this, you can see he's drawn some representative examples of different neurons, and he has these beautiful little arrows, which on the end almost have like in a bow and arrow, like an old-fashioned bow and arrow, you would have at the end of the arrow the little feathers at the end to stabilize the arrow as it flies through the air. He's drawn those in. And so you can see an example of a neuron that sends its axon, which is the extension that goes down and connects with the downstream neuron. And you can see these arrows indicating what he is interpreting as the direction of information flow. And some of this figure, so it's a very, a very prescient and observation and speculation, because at that point, it took many years to prove that information does flow in this direction. Now, some things in this picture are wrong, but many things have laid the groundwork for neuroscience today. And what he's essentially pointing out is that information flows through circuits, neurons interact to guide behavior. So you see something, it enters through your eyes, flows around through neural pathways, and then eventually gets to the motor system that allows you to execute a motor act in response to whatever you saw. Now, this is sort of a hardwired kind of approach. Information flows through, bounces around, and elicits an output. And so the question, comes up now: can memories be stored in something that is fixed and immutable? Can you store information in something that doesn't have the ability to change? And I will say emphatically, no, we know this now. But even back then, there were other, here we have psychologist Ribot, who was thinking about memory as a biological phenomenon. And he writes in his book, Diseases of Memory, every impression and he's talking about just biological material, organic materials, and physical materials in general, every impression leaves a certain ineffaceable trace. That is to say, molecules once disarranged and forced to vibrate in a different way cannot return exactly to their primitive state. If I brush the surface of water at rest with a feather, the liquid will not take again the form which it had before. It may again present a smooth surface, but molecules will have changed places, and an eye of sufficient power would see traces of the passage of the feather. And based on this work, we still use today the word memory trace. We say that when something happens and a signal enters the brain, it leaves a trace. And what he's saying here is that anytime you interact with the environment, you're going to leave a trace. If you walk on the ground outside, which I can see is moist and a little muddy, you're going to compress the ground and leave an imprint. And that is going to change how that piece of ground interacts with the environment in the future. Someone else steps on that part of the ground, the texture will be different. It will be harder in certain points, right? It almost, in a sense, has a memory, so to speak. And he's saying the nervous system is probably the same way. There must be some trace that is left by neurons being active that then allows for the nervous system to act differently in the future, for behavior to be different. And that is the basis of memory he's proposing. Now, it's been about 100 years since Roubaud said these great words. And now, thanks to the amazing technological advances, I mean, it'd be really nice if science moved faster. It is a slow process. And sometimes you look at what's happening and it moves really fast, but it's taken 100 years to really see the plasticity that he's talking about. And so with modern methods, we can see this very clearly. Now, we have, for example, on the left, a little just short video showing one of these processes coming out of a neuron. And you can see that there are all these little smaller extensions coming off, attempting to connect with other neurons, and some grow and retract over minutes, searching out potential partners in the neural network. On the right, we see an example of still images of a neuron over the course of 8 days in the living mouse. And you have the extensions that go out and attempt to connect with other neurons, the dendrites, and these little protrusions coming off of it are called spines, and they specialized structures that receive inputs from other neurons. And you can see, for example, the yellow one, the one indicated by the yellow arrowhead, is present on day 1 and day 2 and all the way through to day 8, so very stable. But you can see other examples like this one indicated by the red arrowhead that appears on day 5, it's there on day 6 and 7, but on day 8 it's missing. Another one appears on day 5 out of the blue and is gone on day 6. So there are all these structures coming and going, attempting to communicate and connect with other neurons. Some are stable, probably get involved in a network. Others are maybe temporary and have a transient function. And maybe others attempt to form a connection and don't, and maybe they never did anything. And so this has really been a beautiful demonstration of how dynamic and ever-changing the brain is. And these things can all be modified by experience. Okay, so we've concluded it's clear plasticity exists. Neurons can change their form and their structure. Now, how do they work together to store memories? We know that they work in a network to guide the flow of information and influence behavior, but how do they store something? Okay, and so to illustrate this, we're going to look at an example from my life, the personal experience that I've gone through. And we can look at 5 hypothetical neurons in a network or in a brain region, such as the hippocampus. And each neuron in this case represents a distinct piece of information. Okay, so what do we have? We have neuron number 1 is represents a former lab member in my lab named Rachel. Great student, always cheerful, did great work. I miss her dearly. And another neuron, neuron number 4, represents bolero carrots. These are my favorite carrots to plant in the garden. They're sweet and crunchy. Number 5 is speed stick deodorant, specifically active fresh scent speed stick deodorant. And neuron 3 is an AO860 microtome, a probably 50-year-old piece of equipment built like a tank, and one of the best ways to cut brain tissue into thin slices. And number two is a grain of sand. I just chose that because I didn't have much space to put something else in there. And so I chose a grain of sand. So we've got 5 neurons that represent 5 different things in the brain. And these are clearly all seemingly unrelated, right? None of you have a association between any of these items. And in my brain too, at one point I had knowledge of all these. There were neurons that represented each of these pieces of information, but they weren't connected in any important way. Now, one day I had an experience. I went up to the lab and I saw Rachel. She was cutting brain tissue on the AO 860 microtome. And I went up and talked to her, and I was a little self-conscious because normally I wear a speed stick deodorant, but I wear the ocean surf scent. That's my scent. I find it kind of unobtrusive, not overly pungent in its odor. And I must have made a mistake because I bought Active Fresh, which is a similar color. It's also a bluish green kind of color, but the odor is much stronger. And I felt like I was walking around like a cloud of perfume. And I was a little self-conscious. And so I went up to talk to her and I said, hey, look, if you smell anything, it's just Active Fresh. I bought the wrong one. I tried to put on not too much, but hey. And she thought this was hilarious that I would come up and I would divulge my underarm habits to her there in the laboratory. And so it was quite a memorable event for a number of reasons. And so what happens? Well, you've got one neuron that represents the AO 860 microtome, number 3, being active, because that's a part of the event. My Rachel neuron is active, neuron number 1, and my speed stick Active Fresh neuron is also active, neuron number 5. And so there's a principle in neuroscience where neurons that fire together wire together. That is, neurons that are connected to each other, like in a sort of potential latent sort of way, if they are active together, the connection between them will be strengthened. That's the condition. And this was proposed by a pioneering neuroscientist from McGill University, Donald Hebb, and it has proven to be true. And so here you can see that all the 5 neurons here have a potential connection, maybe a weak connection between them, but only a subset, neurons 1, 3, and 5, that represent these distinct pieces of the experience, only those ones are highly active, because these are the items that were present in my experience when I met with Rachel and had the Active Fresh deodorant. And so because they're active, you can see the speed stick neuron is going to activate the AO 860 neuron, and the AO 860 neuron is going to activate the speed stick neuron. And because of their mutual activity, the connection between them is now going to be stronger. And likewise, in terms of their connections with Rachel. Now, grain of sand, that neuron was not active. My carrot neuron was also not active. And so even though they may have had some weak potential connections with these other neurons, because there was no activity in those neurons, they didn't strengthen their connection in this subnetwork of neurons. So neurons 1, 3, 5 are now joined together structurally and functionally, to the point that now I can go home, I can, make dinner, read a bedtime story to my children, and this network is not being used. I'm not recalling this memory, but there is still an enhanced connection between these neurons that's present. It's not active at this moment, but there have been structural changes. There's been neuroplasticity that has bound these neurons together, such that at a later date, if I go to Shoppers Drug Mart and I'm picking out deodorant and I set my eyes on Active Fresh Speed Stick, I may activate that speed stick neuron, which will in turn, because of its preferential connections with my Rachel neuron and with the AO 860 microtome neuron, cause a full recapitulation, reactivation of the full network, so that now I can recall that memory in all its glory, essentially, because these neurons are now strengthened, their connections between them. And so that's how we can think about the significance and importance of plasticity. It allows for information to be linked together such that you can recall an experience in all its glory, or embarrassment, or whatever the outcome or significance of that experience may be. Okay, so I think with that as the background, now we can take a moment and think about, well, how might neurogenesis fit into this? And what is the significance of neurogenesis? So, you know, I've written here that the ultimate form of plasticity is neurogenesis, and it continues in the brain's memory centers in adulthood. And really, I don't know if it's the most important form of plasticity, but it is a significant one in that if you're going to add an entirely new neuron, that means you're going to add a whole set of new connections, right? And if memory is made in the connections between neurons, then by adding new neurons, you're necessarily going to be increasing the number of connections you can make, right? Which would seem to be pretty important for memory. And so what is the part of the brain where it's added? We've talked about it being in the hippocampus. It's in a subregion called the dentate gyrus. And so we have this region shown here as this C shape. And I've illustrated two neurons here. One is an adult-born neuron. So this would be, one that could have been born in your brain yesterday, or maybe more likely a few weeks ago, because it has a little bit of a mature sort of appearance to it. And then you're also going to have some neurons that were born in the neonatal period, maybe in the days after you were born, maybe when you're still in the womb. And so there's many different neurons of different ages in this structure. And for here, I've just shown an adult-born neuron and a neonatal-born neuron, just to provide a bit of contrast. And so if we think about the part of the brain that they're added in the hippocampus, it's useful to think, well, what is coming into the hippocampus? Like, what really makes up our memories for everyday events? And the signals that come into the hippocampus come from a region called the entorhinal cortex. And the entorhinal cortex receives very high-level processed sensory information. And specifically, you could break it down into two broad classes of signals. One that carries information about places. And so you can see these little circles represent neurons that represent certain places, and they send axons to the new neurons and maybe the older neurons to connect with them. And you also have a subset of this region, subset of the entorhinal cortex, that has information about the details of an experience, the things that were there. This would be like in my previous example, the place would be the laboratory I was in, the room. I can picture that room. But then the details were, well, they were the deodorant. They were Rachel. They were the microtome. There was the chair in that room, in that part of the room we were sitting in. And so that also gets sent to the hippocampus, to this area where the new neurons are added. And so the critical thing here, the conceptually important thing, is that you can see that these adult-born neurons receive both information about the place and the details. And if you think about it, places and the details of what was in that place, those two things together are what define an experience as unique, right? You may have had multiple experiences in the same Safeway. Maybe you're in the checkout line, and maybe one time the cashier tells you a funny story and you share a laugh. Another time you could have been in the same checkout aisle, but maybe you had a lot of groceries and you dropped one and you had a very different experience. Now, you were in the same place, but the details were different, right? So every experience has a unique set of details, even if it's in the same place. And so essentially what I'm trying to say is that these adult-born neurons are added to a part of the brain that is critical for defining unique experiences and potentially remembering them. And so this is our cartoon example, and we can see now an example of what it really looks like when we label these newborn neurons. So we can look at the same picture from in the next slide here. And so what you see, each of these little dark dots is the cell body of a newborn neuron, and they send their so-called dendrites up this way, and that's where they receive that information about places and details of experiences. And they integrate that, they form some association, and then they send that through their axons, they send that signal to downstream regions to store memories and to process these memories and to use them for various behaviors. So they're added in this critical part of the circuit. If we look at one neuron, it looks like this. This would be the area where the incoming signals about places and details are arriving. If we zoom in on one branch of these dendrites, we can see all the little spines, which are those structures that receive inputs. So this spine might receive some information about a detail of the environment. A neighboring one might receive information about the place. And on the output end of these neurons, when they convey their signals to downstream circuits, they have these really large, some of the most enormous synaptic structures in the brain. These very large presynaptic terminals that release neurotransmitter that then activate downstream neurons. And so this is the structure, but during a critical period when these neurons are young, so in mice and rats, this would be like when the cells are 4, 5, and 6 weeks old, these neurons are better at forming connections. They, in response to a relatively weak stimulus that might otherwise go unnoticed, these neurons are better at strengthening their connections, which would suggest they're more likely, they're more likely to be recruited into memory networks. And a number of things can change the development and the plasticity of these neurons. For example, learning, if it happens when the neurons are a certain age, learning will actually rescue these cells. So many are produced, but only about 20 to 50% of them survive. So an excess is produced, but learning will rescue some of these cells. And learning will actually kill some others. So learning and enriching experiences essentially refine the network and select some to be rescued to store information, and they allow others to dwindle away that maybe didn't store the right information or didn't do something relevant and are not needed. And interesting, some things that do not seem to involve learning or really enriched environment sort of stimulation can also alter these new neurons. For example, exercise dramatically boosts the birth of newborn cells. And it also increases their survival, makes them more likely to integrate into these memory networks. I don't know if that means exercise on its own would make you smarter, but it will potentially increase the potential for these neurons to be there so that they can be used in a learning situation. And so a lot of experiments have been done to see if these neurons are important for memory. And a large number have found that indeed they do make an important contribution. So I've just shown here a couple examples of the types of experiments that are done to test the role of new neurons in learning and memory. And these are classic tasks that are used to study memory in mice and rats. Most of this is drawing on mice and rats. We'll draw connections to human significance, but obviously it's very difficult to study neurogenesis in humans. Not to say it hasn't been done, but most of the work comes from animal studies. And here on the left, we see a famous task called the Morris water maze. And essentially in this task, a mouse or a rat is placed in a pool of water that has opaque paint added to it. So the animal cannot see that there's a hidden platform that's slightly submerged under the water. And so when you put the mouse or rat in the pool, it will swim around aimlessly until it happens upon the platform by chance. And after a number of trials, it will, once it reaches the platform, it will essentially learn the relationship between the different stimuli in the room. And remember, that's essentially what memories are. They're drawing connections and links between all of the different elements of an experience and how they relate to one another. And so with training, you can put the mouse or rat back in the pool, and it will essentially, by looking at the cues on the walls, make a beeline right for the platform. And neurogenesis isn't so important to learn this task on its own. It's more important for really the high precision type of learning, the precise search for it, the platform, and the ability to find the platform if it suddenly changes locations, suggesting that new neurons may be important for storing the details of where it was before, where it is now, making these memories distinct so that one can behave flexibly when there are multiple scenarios that are competing with one another. Likewise, another test that's very common is to study memory for different contexts that may bear a lot of similarity to one another. And so often you can test this in a contextual fear conditioning chamber. And so here, a mouse or a rat would be put in this box and given an aversive and painful foot shock, but not a shock that will hurt the animal, but it's just unpleasant. And when it's put back in this chamber, it will elicit this defensive freezing behavior, essentially freezing in anticipation that this is an environment that is dangerous. And good hippocampal memory will enable the mouse or rat to freeze in the place where it received a shock, but not in a related environment that is safe. And so because neurogenesis is thought to then be important for forming and remembering these details of experiences, animals without neurogenesis usually have deficits in this type of test. They're unable to distinguish harmful places from safe places because there are a lot of similarities between these two environments. They get easily confused. But it's not just memory. Neurogenesis, interestingly, is also important for mood and reward-related behaviors. So on the left, I have a picture of a simple anxiety type of behavior test. Here, it's called the novelty suppressed feeding test. And this shows a picture of a mouse in a new environment that it's never been in before. And if you put a mouse in a new environment, it usually is a little nervous. It's a little anxious. It's not sure what to expect. And so its tendency is to stick around the perimeter of the environment near the walls where it might be safer. If it ventures into the middle of the chamber, I mean, who knows? A bird could swoop down, right? And get it. So now what you do is you put a little bit of food in the middle that entices it. So it wants to go to the middle, but it also knows that it's going to be vulnerable in the middle. And so that induces this state of anxiety where it wants to investigate, but it's also scared to investigate. And so, you know, everyone's different where they draw the line in this sort of situation. And if you eliminate neurogenesis from a mouse, you'll find that they're less likely to venture out into the center and get the food. They're more cautious, they're more anxious, they're more innately fearful. And this test is interestingly sensitive to antidepressant drugs like fluoxetine, Prozac. And so if you give a mouse Prozac, it will go venture into the middle of the chamber more quickly, get the food. But if it doesn't have neurogenesis, it will not. So the antidepressants require neurogenesis in order to work. At least in this task, not in every behavior, but at least in that task. Another experiment that we've done is shown here on the top right, which is pretty interesting and unusual in that it also doesn't seem like a behavior that would really depend on memory. And it's essentially a task where we give a rat a choice between one lever, the low reward lever here, where if it presses that lever, it will get a sugar pellet and they'll get it right away. So that's pretty good. But on the other hand, it can press the high reward lever and get 4 sugar pellets. And so naturally that would seem like a better option, but the issue is it has to wait for these 4 sugar pellets. It has to wait 10, 20, sometimes 30 seconds to get the reward. And you can imagine if someone says, you know, do you want $10 now or $10 in a week? You know, you're going to take the $10 now. But what if you are offered $10 now or $100 in 2 months? You know, then it becomes a real conflict, right? You could do something now with the $10. Do you trust these people? Are they really going to give you the money later? You know, how impulsive are you? How much do you want it? But if you stop and think about it, maybe you realize, well, I can wait. $100 is a better option. And what we find in this sort of conflict is that rats that don't have neurogenesis are more likely to choose the immediately available reward. They're more impulsive. And we don't know exactly why that's the case. This is sort of a new line of research. But it is interesting that the hippocampus, this part of the brain involved in memory, it seems it's important for forming representations, not just of the past, but also of the possible future. it seems like it's needed to just form memory-like representations, regardless of whether it's in the past or the future. And so you can imagine that if the rats don't have neurogenesis, they're not as able to form, accurate, detailed representations of the world, then maybe they can't quite envision or predict or imagine the future reward, what might be happening in 30 seconds when they press this lever, that they might be getting these 4 sugar pellets. That's one possible explanation for this type of behavior. And the interesting thing there is that this type of behavior, this impulsivity, is disrupted in many conditions. Addiction is clearly disrupted in schizophrenia. With Alzheimer's, it's affected as well, and also in depression. So it's a very, you can see it even in children are more impulsive. So it's just a behavior that is common across all individuals and is relevant in so many situations. And then I think the last one I'm going to talk about is neurogenesis buffers the effects of psychological stress. So here we have an experiment, and the data, it just shows the amount of corticosterone, which is the rodent equivalent of cortisol, the major stress hormone, after a psychological stressor. And so you can see that immediately after the stressor, whether you have a normal mouse or one that doesn't have neurogenesis, they're both similar. But 30 minutes later, when the stressor is ended, the animals without neurogenesis have higher levels of stress hormones. And so that is interesting because memory, it turns out, this part of the brain, the hippocampus, regulates the stress axis and regulates the production. It actually regulates how well you turn off the stress axis. And so it does, when you think about it, make intuitive sense that the way you react to situations in the world is going to be dependent on what you've gone through in your life. And the hippocampus, as something that stores memories of the past, is therefore in a position to control how likely you are to get stressed out by something based on what you've gone through. So neurogenesis is figuring into this, and seems to be important for suppressing the stress axis, preventing it from getting out of control. Okay, so I have just a few more slides. We've looked at the behavioral properties and functions of neurogenesis. Now, I think there's another interesting question that's emerging, which is the fact that neurogenesis declines with age. Naturally, there's more neurogenesis in infancy, in the embryonic period, and neurogenesis continues throughout adulthood, but it definitely declines. And so what does that mean? Is that a problem, or how do we interpret that? And so that's a question that my lab has been really interested in. We're interested in how neurons are different, whether they're born in adulthood or development or aging, and how does this affect health throughout the lifespan? And so here you just see a picture of all the neurons that are born on the day of birth in a mouse on the left, and you see a different picture showing neurons born in adulthood. And clearly, this is a young adulthood. And so there are definitely many fewer in adulthood. And so if there aren't as many added in adulthood, and if in aging, even fewer are added, well, is this significant? Can they be doing something if they're not there in terribly large numbers? And so what we began to look at is the long-term development of these cells. When I talk about development, I'm talking about them growing into the network, which in itself is a type of plasticity. And so, as I mentioned before, new neurons have a critical period when they're 2, 3, 4, 5 weeks old. And most people have studied neurons of that age. At this point, they're growing a ton. They're very plastic. There's a lot of exciting things happening, but people have kind of ignored the older neurons. And here we can see a 7-week-old neuron, which is older than most people have studied, and in mice and rats. And we see that some of these dendrites, these processes that receive the information about places and details of experience, some of them are not very long. They don't extend all the way out as far as some of the other dendrites, suggesting that this neuron still has room to grow. And so we looked at neurons that are as old as 24 weeks in the rat, And we have the data shown here on the graph, which shows the length, the total length of all of these dendrites, which is, like a proxy for how much space is available to receive incoming information. And we see that there is the usual pattern of growth when the cells are young, 4 to 7 weeks. But even until 24 weeks, there's considerable additional growth of these neurons. And ultimately, you can see that they reach a length of about 3,000 micrometers. And 3,000 micrometers is 3 millimeters. So that's, you can see that with your fingers, right? It seems pretty small, right? But if you think about the fact that in the rat alone, you've got 2 million of these cells in total, 2 million times 3 millimeters ends up being that there's kilometers of dendritic material that is able to receive, information to store memories. So in humans, there's going to be way, way more. So they grow for a long time. This is about 24 weeks, 6 months is about 25%, maybe more, of the animal's lifespan. We know that maturation is slowed in longer lived organisms. And so in humans, if we were to scale things similarly, it's likely that neurons in humans, the new neurons that are born, mature probably over decades. And interestingly, there is evidence in support of this. Old work that just looked not at newborn cells, but just neurons in the same brain region, found that when you took postmortem tissue from human brains, that these neurons they were larger and larger in the older subjects. And this went up until around the age of 80. And then once people reached very old age, like in the 90s, these neurons shrinked a bit, they got a bit smaller. But it just showed that there is continued growth throughout almost the entire lifespan. And so even if there aren't a lot of neurons being added, there seems to be some reserve of plasticity because they grow for such a long time. And if we zoom in on those dendrites and we look at those spines, those little structures shown by the arrowheads here that are the sites of incoming information, we see that the young neurons that are only 2 weeks old are very thin. They get thicker as they develop and become larger and grow. And you can see, if you look at the 7-week-old neuron and the 24-week-old neuron, the number of spines, potential synaptic connections, is very dense. There are many of them. And it turns out that there are more of them on these adult-born cells than there are on the neurons that were born in the neonatal period, like when the when the animal was born. And we can see that quantified here. For example, the mushroom spines are these large spines that are thought to be particularly important for memory. The number of them goes up and up and up when the cells go from 2 to 4 to 7 weeks. But by 24 weeks, they continue to grow even larger, or even more, and become more numerous. And the number is about double that of those in neurons that were born in infancy. And this is really interesting because, again, there is longstanding work looking at other mammals and primates and even human brain tissue, showing that there is a population of neurons in this part of the brain that have about twice as many spines as the general population. And no one knew what to make of this. And so I think our data would suggest that these adult-born neurons, or the neurons born later in life, simply have more connections than the other neurons. Okay, so even if neurogenesis declines with age, what our research is finding is that their effects on plasticity are substantial. So here we have some data just generated from a simple mathematical model that estimates how many cells are added in adulthood. And that's shown by the red line. And so here we have the rat age in days. So this is essentially across the full lifespan of a rat. But presumably, this would scale similarly for other mammals, including humans. And the accumulation of these adult-born cells is shown in red. So more and more and more accumulate. And that's something that's often missed in most neuroscience studies of neurogenesis, that when people are looking at neurogenesis in adulthood, they typically are looking at a snapshot at one moment in time. And they forget that a small number of cells may be added on that day, but the next day some more added, the next day some more added. And over the course of years and decades, that really adds up. And so in this situation, our neurogenesis actually ends shortly after middle age here. And I think that's an underestimation, but that's just the way the model turned out. And so you can see it plateaus. The accumulation of adult-born cells plateaus there. And they end up making about, we think, make up about half of all of the neurons in this part of the brain. So that's quite a bit if you consider the cumulative addition. Now you consider that these cells are growing over 25% of the lifespan. If we look at the number of spines, and especially the number of these large mushroom spines, which are sites of really strong connections with other neurons in the brain, you can see that the numbers of these mushroom spines continues to increase all the way until the end of life, even though there might not be many cells added at the very end of life. The continued development of these neurons allows for there to be an addition of plasticity up until the end of life. So we think their effects on plasticity are substantial. And then, this just speaks to a number of ways in which neurogenesis might be harnessed to promote mental health in adulthood. So I'm just going to briefly highlight 3 examples. Well, one, if we look at the cartoon example of the entorhinal cortex sending information to the hippocampus, we know that in aging, memory changes, right? Some forms of memory actually get better. There's semantic knowledge and wisdom, and that comes from experience, that comes with age. But some of the memory related to specific experiences, becomes impaired, and people may not be quite as good at it as they used to be. And what you see is that in aging, there's a pathology related to Alzheimer's disease, but it actually is happening in all of us in adulthood. And whether it becomes a real problem or not, it's a bit of a mystery why that happens. But this shows the staining for the histological staining for a protein called tau, which ultimately becomes toxic in the memory centers of the brain. And this is why memory is one of the first, cognitive functions to become impaired in aging and in the earliest stages of Alzheimer's disease. And you can see where it first appears is in the entorhinal cortex area and the hippocampus. And specifically, the first area that seems to be disrupted is this area that sends information about the details of experience to the hippocampus. And so if new neurons are being added to this circuit where they're receiving these signals about the details, there's a possibility that adding plasticity to this circuit could, prolong its functionality for just a little bit longer, allow those types of information to enter the hippocampus more effectively if neurogenesis could be harnessed. So it's relevant because it's occurring right in these sensitive circuits in aging and Alzheimer's disease. I already talked about things like preferring delayed rewards that may be better. Well, it's been shown that if you envision the future, and that can actually allow you to make better decisions and make choices that may be better for you in the future and are not immediately gratifying. And so these types of behaviors and choices are disrupted in disorders like depression. And I also mentioned schizophrenia, but there's other functions related to reward, where rewards are, how much are you going to work for them? A lot of these things depend on memories for where you've gotten rewards before and how you've gotten them. And so neurogenesis has a role to play in treating conditions like depression. And then lastly, we talked about neurogenesis being important for being able to distinguish different places, and also being important for regulating anxiety types of behaviors in animals. And so this becomes important. In this example, you can see there might be a soldier that has returned from war and is seeing a cue that might remind them of, traumatic experiences that they've gone through. And so part of this, can be adaptive. If you see something that reminds you of something bad that happened, well, it can be good to be on edge, to be ready, right? But on the other hand, if you're clearly in a safe environment, you don't want to recall those adverse experiences and apply that memory to the current situation. You want to keep these experiences distinct, so that you're not essentially stressed out unnecessarily, right? And so where you draw that line is going to be influenced by neurogenesis. So neurogenesis has the ability, by enabling us to better remember experiences and to be able to remember the details and keep them distinct. It might help us to display anxiety and fear behaviors in an appropriate setting and not in excess, essentially. So that's just a bit of a snapshot of neurogenesis and plasticity and why I think it's important. Obviously, I've been able to do this with a lot of help from members of the lab and funding agencies like the Canadian Institutes of Health Research, the Centre for Brain Health here at UBC. And I'd just like to thank you for listening, and I look forward to hearing any questions that you have.
Awesome. Thank you so much for sharing your expertise with us, Jason. I find it really interesting, and I'm sure everyone else as well, very exciting to hear about how our brain is changing and growing day to day, and even at this very moment, potentially. It's easy to see how this can apply in terms of brain function, and interestingly in Alzheimer's and mental health as well. We do have a few quick questions before we finish off. One attendee was wondering, what promotes the creation of these new neurons in adulthood?
Right. So I mentioned the most robust one is exercise. Exercise is one of those things that you were always told you can't go wrong with exercise, and it turns out that it increases the birth of these cells. So you have stem cells in the hippocampus. I can unshare my screen, I suppose. You have stem cells in the hippocampus that divide and give rise to newborn neurons. And so exercise will cause them to divide at a more rapid rate. So they basically have more daughter cells. So you're going to have more new neurons. Now, I mentioned that also these neurons will die if they are not integrated into the circuitry. And so exercise will also rescue them as well. I don't know that it will work forever. It works for a while. I'm not saying don't keep exercising, probably, but to some extent, you can't keep adding neurons indefinitely, because you have a skull that is a fixed volume, and we can't just keep adding these cells because we have limited space to put them. Something's got to give, right? But enriched environment learning will rescue some cells and remove others. Things like inflammation, things that certain immune responses can suppress neurogenesis. The list is endless, because the receiving inputs from the sensory world, they have receptors for stress hormones and other hormones. And so many things can modulate it. In a human that's behaving in a complex world, all of these things interact. And so, but in a controlled environmental setting, studying animals, you can give certain dietary things and maybe see changes, for example. So they are very sensitive.
Awesome, very cool. And it's interesting to hear, especially about exercise, as it's obviously very relevant to our program here at Brain Wellness. Another question that came up was that you discussed how rats without neurogenesis looked more for an immediate reward, and how this may relate to addiction. Do you think this might translate to an inherent susceptibility of certain people for addiction or potential therapeutics in the future?
Yeah, I think, I think new neurons would be one part of a circuit involved in this behavior related towards reward seeking. And so, if something may have happened to an individual, or there may be some genetic predisposition that causes neurogenesis to be reduced, they may be more biased to be choosing, things that are immediately gratifying, and less able to hold off and envision those rewards that would come in the future. So, I mean, there's definitely so much variability we see in all individuals, right? And so this could be certainly one factor. So
Awesome.
maybe
I'll just
promoting neurogenesis would counteract that. We actually tried that. We tried that by we gave rats running wheels to boost neurogenesis, and they were less impulsive. They were more likely to wait and choose the delayed reward. The effect was, I believe it was there, but it wasn't statistically significant. We probably needed a larger pool of subjects to conclude definitively. But that made me believe that there are ways of actually counteracting that, and exercise would be one. Yeah.
very cool. Awesome. So I just want to thank everyone so much for coming today, and especially thanks to you, Jason, for this excellent and very interesting presentation and answering these questions for us.
My pleasure.
I've really enjoyed learning about neurogenesis alongside you all, and I hope you will also join us again on May 3rd, where we will be celebrating Women's Health Month by learning about sex differences in age-related brain and muscle function. And you can find more info and register for that event on our website under the events page. And lastly, before we sign off, if you are enjoying the program and events like today's research seminar series, please consider making a donation through the UBC Giving Day site. Our website has a banner at the top of our homepage with a direct link to our donation page, and that link can also be found in the chat. In the last hour, we have increased donations to $35,000 from 103 generous people, and are quickly on our way to reaching our goal of $50,000 to support our program. So thank you all for your support and joining us today and spreading word to your family and friends. And one last big thank you to our wonderful speaker, Dr. Jason Snyder, and all of you for joining us today. So please enjoy the rest of your Wednesday, and have a great afternoon, everyone. Goodbye.